In this video, we will explore the processes of photosynthesis in detail for the higher level. We will look at the structure and role of the chloroplast, the role of Photosystems I and II, the light-dependent reactions, and the light-independent reactions, otherwise known as the Calvin cycle. Let's start by thinking about what the definition of photosynthesis is. Photosynthesis is the process by which photoautotrophs, including plants, algae and cyanobacteria, use light energy to fix atmospheric carbon dioxide into carbohydrates. So they convert light energy into chemical energy stored in the bonds of carbohydrates. This is the overall equation for photosynthesis. Photosynthesis is actually made up of a series of reactions known as the light-dependent and light-independent reactions. In plants, photosynthesis takes place in the chloroplast. The chloroplast is a membrane-bound organelle. Although this is covered in the standard level key concept video of photosynthesis, we are going to be talking about these structures when going into the details of the reactions, so we should go over these first. If we look at this diagram of the chloroplast, we can see the main structures. Here we have the inner and outer membranes from the double membrane or envelope around the chloroplast. The double membrane is believed to be evidence of the origin of chloroplasts. It is thought that chloroplasts evolved from photosynthetic bacteria, which were engulfed by larger cells about 600 million years ago. Because they were engulfed, the outer membrane of the chloroplast was formed from the vacuole membrane during endocytosis. The engulfed bacteria were not broken down, so they continued to photosynthesize, producing organic molecules, which the host cell could use, and the host cell provided protection, so it was a symbiotic relationship. This theory is therefore called the theory of endosymbiosis. This is why chloroplasts have DNA and the smaller 70S ribosomes that prokaryotes also have. You need to know about this theory. Carrying on with the structures then, this is the stroma, which is the fluid-filled region inside the chloroplast, which is a site of the light-independent stage of photosynthesis. Here are the thylakoids. These form the grana, a series of flattened sacs stacked on top of each other, and the intergranal lamellae. These lamellae ensure that the grana work together as one unit. The thylakoids are the site of the light-dependent reactions of photosynthesis. The photosynthetic pigments are found in the photosystems embedded in the thylakoid membrane, which absorb light energy for the light-dependent stage of photosynthesis. Starch granules and plastoglobules, lipid droplets, are involved in storage and metabolic processes. The key adaptations of the chloroplast for photosynthesis are: the thylakoids provide a large surface area for the absorption of light, and the thylakoid space is very narrow, allowing a rapid build-up of an electrochemical gradient for chemiosmosis. More on this very soon. The thylakoid membranes contain the photosynthetic pigments responsible for the absorption of light energy, which is converted into chemical energy. The thylakoid membrane also contains the electron carriers for the electron transport chain, the details of which we will be going over later. The stroma contains the enzymes necessary for the light-independent reaction. The thylakoid membranes are surrounded by the stroma, so the products of the light-dependent and independent stages have only a short diffusion distance to move between the two. Now that we have looked at the structures of the chloroplast and highlighted where the light-dependent and independent stages occur, let's look at these stages in more detail. We will start with the light-dependent stage. This process involves the photosystems I and II. These photosystems are found embedded in the thylakoid membrane. They are made up of proteins and photosynthetic pigment complexes. The photosynthetic pigments include xanthophylls, carotenes, and chlorophylls A and B. The reaction centre here just contains chlorophyll A, and the absorption of light by the pigments generates excited electrons from chlorophyll A in both photosystems. Although they both contain photosynthetic pigments, the optimum absorption of light energy of their primary pigment is at different wavelengths. Photosystem I absorbs light at a wavelength of 700 nanometers best, whereas Photosystem II absorbs it best at 680 nanometers. Just to note, the photosystems are called I and II based on when they were discovered. It has nothing to do with the order of reactions, as these occur simultaneously. Now let's look at the importance of these photosystems. The photosystems are involved in what is sometimes called the Z scheme because of the shape of the diagram explaining it, which you can see here. At the start of the Z scheme is Photosystem II. The photosynthetic pigments in Photosystem II absorb light energy, and this energy excites electrons from chlorophyll A in the reaction centre, a process known as photoactivation. A pair of excited electrons escape from the chlorophyll A, oxidising the chlorophyll A, and pass to an electron acceptor in a higher energy state, thus entering an electron transport chain, shown here. The electron acceptor has gained electrons and so is now reduced. The reduced electron acceptor that has just received the electrons from the chlorophyll then passes these electrons to the next electron carrier, thus reducing the electron carrier that is receiving the electrons and oxidising the electron carrier donating the electrons. This process of reducing one molecule while oxidating another is known as a redox reaction. As the electrons are passed from electron carrier to electron carrier along the chain, energy is released. The energy is used by the electron carriers to pump protons from the stroma into the thylakoid space, building up an electrochemical gradient. Chemiosmosis now occurs when the protons diffuse out of the thylakoid space down their electrochemical gradient back into the stroma through special proteins in the membrane called ATP synthases. The movement of the protons through the ATP synthase drives the phosphorylation of ADP with inorganic phosphate to form ATP, seeming as the electrons that provided the energy to pump the protons into the thylakoid space gain their energy from light, and the chemiosmosis of the protons through ATP synthases resulted in the phosphorylation of ADP to form ATP. This whole process is called photophosphorylation. This ATP is then used in the light-independent reaction. The de-energized electrons are then passed to Photosystem I. As light energy is absorbed by pigments in Photosystem I, a pair of electrons are again excited and pass to another electron acceptor. Some of these electrons may return to the electron transport chain in cyclic phosphorylation to produce more ATP, or the electrons may be transferred to carrier proteins, which then pass the electrons on to NADP, and reduced NADP is produced. So, as we can see in this diagram of the Z scheme, Photosystem II is only involved in non-cyclic photophosphorylation, whereas Photosystem I is involved in both cyclic and non-cyclic photophosphorylation. Photolysis of water is an important reaction to remember. It is often forgotten about when students answer exam questions, but the light-dependent stage could not continue without it. Photo means light, and lysis means to split. So photolysis of water molecules is the process by which light energy splits the water molecules apart. This gives rise to protons, which are used in chemiosmosis and in the formation of reduced NADP. Electrons, which replace those lost from chlorophyll A in Photosystem II, and oxygen, which is released as a waste gas. So looking at the two types of photophosphorylation, cyclic photophosphorylation is when Photosystem I only is involved. ATP is made, but there is no reduced NADPH. In non-cyclic photophosphorylation, both photosystems are used, and electrons reduce NADP to NADPH. In photolysis, water is split to provide electrons for Photosystem II and protons for reduced NADP and ATP production, and oxygen a waste gas. Now let's look at the light-independent stage. The light-independent stage, as I mentioned earlier, occurs in the stroma of the chloroplast. It is the stage when carbon is fixed to form the organic molecules that are the products of photosynthesis. Carbon dioxide is a substrate in the reaction, and glucose is the product. The ATP and reduced NADP, which were produced in the light-dependent stage of photosynthesis, move from the thylakoids into the stroma to be used in the light-independent reaction. During the light-independent stage, the carbon dioxide that is diffused in through the stomata of the leaf combines with the 5-carbon molecule ribulose bisphosphate, otherwise known as RUBP. This reaction is catalysed by ribisco, a carboxylase enzyme, and it results in an unstable 6-carbon molecule being formed, which then splits into 2 molecules of the 3-carbon molecule glycerate 3-phosphate, GP. Each glycerate 3-phosphate is then reduced to triose phosphate, TP. The reduction of each glycerate 3-phosphate molecule involves 1 molecule of reduced NADP and 1 molecule of ATP from the light-dependent phase. So for the 2 glycerate 3-phosphates, 2 reduced NADP and 2 ATP molecules are needed for their reduction. The oxidized NADP can then return to the light-dependent reaction to be reduced again. Some of the triose phosphate is used to form carbohydrates such as glucose. 1 out of 6 triose phosphates are used. The rest of the triose phosphates, 5 out of 6, are regenerated into ribulose bisphosphate, so the cycle can keep occurring, fixing carbon from the carbon dioxide in the air. This order can be remembered by using this mnemonic for the Calvin cycle. Rabbits can get really tired running around. R for ribulose bisphosphate, C for carbon dioxide, G for glycerate 3-phosphate, R for reduction, T for triose phosphate, R for regeneration, and A for ATP used again. It is easy to get confused about how the just 1/6 of the carbon in triose phosphate goes off to produce carbohydrates such as the hexosugar, the 6-carbon sugar, glucose. If 1 out of the 6 carbons from the triose phosphate is used towards making a hexosugar for each turn, i.e. for each carbon dioxide molecule fixed, then 6 turns are required to make 1 hexosugar. So if we put 6 carbon dioxides coming in here, that is 6 carbons. That means we need 6 molecules of ribulose bisphosphate for the 6 carbon dioxides to bind with. 6 lots of the 5-carbon ribulose bisphosphate gives us 30 carbons altogether. They form 6 molecules of the unstable 6-carbon compound, so there are 36 carbons here. These 6 unstable 6-carbon molecules then split to form 12 molecules of the 3-carbon glycerate 3-phosphate, which still gives us 36 carbons. These are reduced to 12 triose phosphate molecules, still 36 carbons. 1/6 of the triose goes off to form carbohydrate. 1/6 of 36 is 6. So here are the 6 carbons to form the hexosugar, leaving 30 carbons to go off to regenerate the 6 molecules of the 5-carbon ribulose bisphosphate, and it starts all over again. And now we've come to the end of this key concept video for the higher level section of photosynthesis. So I'll leave you with a summary of the light-dependent and independent reactions, so you can read through them at your leisure. The key terms are highlighted. Remember to use these in your written exams.